Good morning, and thank you to OIS for welcoming us to present this new data we just got on our phase 2b study in wet AMD. We are a public company, and this is our forward-looking statements. So we work on long-acting formulations for potentially transformative treatments in vision-threatening diseases. And I will be reporting today this 18-month data we got on our lead asset, GB102, demonstrating a year or more of duration for the majority of patients. We work on differentiated clinical programs that are targeting large markets, and we have good IP protection all the way to 2041. We are looking at building a bigger pipeline of novel therapeutics, and we have a fair amount of cash supporting our operations into 2023. Our technologies are injected in the vitreous with a small gauge needle, and from our polymers, drugs would elute over time, the polymers would disappear. And as you can see here, we are targeting differentiated mechanism of action as exemplified with our lead asset. So the phase 2b in wet AMD was the following. We had two phases. The first phase was a 1-year core study where patients that were non-naive patients were enrolled, randomized into 3 groups. The first group was given GB102 1 milligram on day 1, as well as at month 6. The second group was given GB102 2 milligram at day 1, and the repeat dose was done with 1 milligram at month 6. And the third group was a control arm with Eylea given every other month as per label. And these patients were followed up monthly for a year. And the primary endpoint was first time to rescue. There were rescue criteria. All 3 arms could get rescued. The secondary endpoints we're looking at all the traditional Retina endpoints. Patients at the end of this first phase were asked if they wanted to volunteer to enter an observational phase of an additional 6 months. To be eligible, they needed not to have been rescued on the last visit of the core study, as well as being willing to be monitored monthly for this extension period. And roughly 60% of patients volunteer to be part of the extension study. So here are the results for GB102 1 milligram, which is the dose we are pursuing. In the first 12 months of the study, in the core study, we reported half of patients experiencing a 6-month duration of their treatment in wet AMD. With the 6-month extension study now, these numbers have increased, and we now have 55% of patients reporting a year of duration of treatment. The treatment burden in the core study was reduced by around 60%. The treatment burden, when you put the patients in the extension study, is actually reduced now by 75% compared to what patients were getting before getting in the study. The safety, we had a favorable safety profile in the core study. We have the same in the extension study. And the anatomical control, which, as you know, is a very important biomarker in this disease, we had the similar control to Eylea in the core study, and that is also what we've seen in the extension study all the way to 18 months. BCVA was the key surprise in the core study. We reported that we had a BCVA trend that was lower than Eylea in the core study, but we've seen that BCVA maintained in the extension study. So just to focus on BCVA, which is a key endpoint, we are able to explain the BCVA difference between Eylea and GB102 1 milligram by 3 different factors. The first 2 are on the left-hand side of the slide. We had treatment-unrelated AEs or hard-to-treat patients that were, because of the size of our study, sadly randomized into the GB102 1 milligram arm. And we believe these patients, when we do larger trials, will be evenly allocated to a number of to the different arms. On the right-hand side, clearly what was related to GB102 was particle dispersion. As much as we had improved the formulation, we did not improve it enough for the particles not to somewhat disperse in the back of the eye and create this cloudiness of the vitreous, which was hampering the patients from seeing as well as they did on the Eylea group. But we have a clear roadmap for success. First, we want to capitalize on the great safety profile, on the unprecedented duration we've seen with a simple IVT injection of a year for the majority of patients, as well as the great anatomical control similar to Eylea given every other month. We are now currently working on enhancing the formulation, and I will show you some data there that will improve the safety and the BCVA. We will amend our entry criteria to make sure we do not take patients that are not anti-VEGF responders. There is no chance if they are not responding on Eylea given every month sometimes, they will respond to anti-VEGF given in the sustained release formulation. And of course, we'll amend our rescue criteria. 60% of our rescues in the core study were unnecessary, either frivolous or simply because of a drop in BCVA. And we're currently entertaining partnering discussion for this asset. So I wanted to share with you some of the tests we are doing, and it's one of the many tests we are conducting on these formulations. On the left-hand side is the formulation we've used for the Altissimo trial. And what we do, we inject this formulation in cuvettes, and after 10 minutes, we flip them around. We put them upside down. And I will show you videos of what the original formulation was doing. So you see here a tail of microparticles, and you see we can't pick up the formulation with tweezers. It's just like a gel. It's not a solid form. The new formulation we have finalized, as you can see here, leaves far less particles behind. And after 10 minutes, you can actually pick it up with tweezers. It has totally gelled and seems to be performing much better. And we're very happy to report that this new formulation is not changing in any way or form the pharmacokinetics of the drug, which we are very happy with. So this is what our pipeline looks like. We also have a glaucoma asset, and I want just to show you first pictures of our new implants in glaucoma. And you can see here from baseline to day 84, which is the latest time point we have, you can see that these particles stay together and there is no dispersion of this formulation in the back of the eye, whilst remaining very long-acting, as exemplified on the graph on the right-hand side. So we are preparing the IND package, and we are expecting to be in a clinical trial in the second half of next year. Thank you.